Hello and welcome to the webinar today, titled Regenerative Medicine Using iPSCs, Setting Your Research Up for Success, and brought to you by RegMedNet. My name is Martha Powell and I'll be your host for today's event. I'm a digital editor of Future Science Group, publisher of the journal Regenerative Medicine. We're delighted to have Fabrizio Rinaldi and Tim Manning from Bio-Techne presenting for us today. This webinar is sponsored by Bio-Techne. Bio-Techne brings together the prestigious life science research brands of R&D Systems, Novus Biologicals, Tocris Bioscience, and Protein Simple to provide stem cell researchers with high-quality proteins, media, small molecules, antibodies, and analytical instrumentation. Fabrizio Rinaldi is a research and development scientist for stem cell products at R&D Systems, a Bio-Techne brand. Fabrizio has extensive experience working with induced pluripotent stem cells, including reprogramming, expansion, and differentiation. Our other presenter, Tim Manning, is the product manager for proteins at R&D Systems, a Bio-Techne brand. Tim has extensive knowledge of a protein production, quality control, and GMP guidelines. We invite you to submit any questions you may have for our presenters throughout the webinar using the Q&A tool to the right-hand side of your screen. We'll pose your questions to Fabrizio and Tim in a live Q&A session after the presentation and then follow up any outstanding questions offline. Thank you again for joining us today, everyone. Fabrizio, I'll now hand over to you to begin today's presentation.
Thank you very much. I want to, first of all, say thank you again to Martha for the kind of introduction and for the opportunity to share some data that I obtained here at Bio-Techne. Second, I would like to thank all the scientists that are following this webinar, and I hope that the data that I will show you, they can be helpful in your research. Today, I'm going to show you some tools that we use here at Bio-Techne to manage iPS line variability. So I will give you a little introduction about stem cell, and especially on induced pluripotent stem cell variability, and then I will give you an overview about tools to reduce variability during colony selection and to minimize the variability during the iPSCs differentiation. So let's start from a little introduction on stem cell and especially on induced pluripotent stem cell variability. What is a stem cell? A stem cell is a cell that has these two main characteristics: can self-renew and regenerate other stem cells, or can escape the stemness phase and differentiate in terminally differentiated cell line. I like to divide the stem cell in two big groups: adult stem cell and pluripotent stem cell. Both the stem cells can be used in different applications, as we can see from the bullet points. For example, regenerative medicine, drug discovery, toxicity assay. What is important? Important is the quality of your cell line. So you should use standardized protocol to isolate and differentiate your cell line, and especially you should check routinely for the purity of your cell line. Induced pluripotent stem cells were discovered in 2006 by Yamanaka and Takahashi that they reprogram adult cells using particular transcription factor in pluripotent stem cells. Before that time, the only pluripotent stem cell used were embryonic stem cells derived from blastocysts. The pluripotent stem cells can differentiate in all the tissues of our body. Why is so important to prioritize the stem cell quality? Prioritize the stem cell quality is very important to ensure uniformity and reproducibility of your stem cell line and their derivatives. As you can see from this slide, there are different aspects that can affect the stem cell quality. Today, I'm going to be focused on one aspect, the pluripotency. I'm going to show you some tools that we use at Bio-Techne to assess the pluripotency of the cells. So let's start with an overview on tools that we use here at Bio-Techne to reduce the variability during colony selection. Which one are the major causes of variability between iPS and line? I show you in this slide with the bullet point different causes, like for example the source of the cells, the chromosomal aberration, the differentiation capacity. But especially there are two causes of variability during the cell reprogramming. One is the way that we use to reprogram the cells, so transcription factor overexpression or small molecules and transcription factor overexpression, microRNA or messenger RNA. The second one is the delivery method, so how we deliver the reprogramming factor in the somatic cells. They could be, for example, episomal vectors, integration-free, or vectors that integrate the genetic cargo in the genome. At the end, the final result is the formation of the colony. So, which one are the characteristics that we should check to pick the right colony? The easiest way is just check the morphology of the colony. So we know that a good colony has clear edges, symmetric morphology, and could be clean to be picked. But it's not always that easy, so sometimes the cells can overgrow and you have colonies with undefined edges, asymmetric morphology that are very difficult to pick. For this reason, here at Bio-Techne, we like a lot to use antibodies for live cell imaging. These antibodies are designed for direct single-step ICC in embryonic stem cells and induced pluripotent stem cell. They don't affect the cell proliferation and sameness. You can have a very clear colony distinction, and they minimize the inclusion of differentiated cells. As you can see in this picture, I show you two markers, in this case SSA4 for the pluripotency and SSA1 for the differentiated cells. In this picture, I show you some colonies that I picked during my research. And in this case, we don't have the DAPI because DAPI can bring chromosomic aberration in your cell population. And in green, you can see the white arrow indicate the pluripotent cells, and in yellow, the red color, SSA1, indicate the differentiated cells. We use three marker for pluripotency, SSA4, TRA160, and TRA181, and one marker for differentiation, SSA1. But sometimes we don't need all the markers, so we can use individually the 4 antibodies, and we can get the antibodies in different flavors. So you can choose what kind of color you want to use in your research, red or green. So, in summary, for the first part of this talk, I can tell you that picking the right colony is very important. The morphology observation sometimes could not be enough. So, having an help using antibodies for live cell imaging can be a very useful tool without compromise your cell line quality. After colony selection, you should find ways to interrogate your stem cell population about the stemness. So here at Bio-Techne, for example, we use live cell imaging, as I showed you before, or flow cytometry, or multicolor immunohistochemistry. We use Western blot analysis as well, and antibody-based multiplex arrays. In this slide, as you can see, for example, we have some pluripotency markers that we use to stain our stem cell population. On the left, I stain the cells with the alkaline phosphatase in red and NaNOG, a nuclear marker, in green. And in the right panel, I stained our stem cell using O34 as nuclear marker in red and SSA4 as a cytoplasmatic marker in green. Another tool that we have in-house to check for marker expression analysis is the Simple Western. Simple Western is a Western blot alternative, and in this slide you can see a Simple Western analysis of 3 time points during the differentiation of iPS cells in hepatocytes-like cells. In the first panel, the cells show a very nice signal for OCT34 at day 0, a pluripotent marker absent at day 14 and day 21. In the other panels, we detect a hepatocyte-specific marker between day 14 and day 21. Simple Western is a capillary-based immunoassay that allows you to separate your protein samples by size or charge. The sample is loaded into a capillary automatically and run by the machine. The separated proteins then are immobilized and identified using a primary antibody, an HRP-conjugated secondary antibody, and a chemiluminescent substrate. The chemiluminescent signal is then detected and quantified, as you can see from the peak on this image, and the images can be exported in a Western blot-like picture. I like a lot this technique, and we use a lot this machine here at Bio-Techne because the process is fully automated, it takes 3 hours, it is quantitative, and you can run 25 samples in one plate with no cross-contamination. The only issue is the primary antibody that should be tested for Simple Western, but here at Bio-Techne we have more than 2,000 antibodies tested for Simple Western. At this point, I selected the right material, I can be consistent with my research, and I can start with my stem cell differentiation. In the last part of this talk, I will give you an overview on tools that we use to minimize the variability during iPSCs differentiation. Which one are the pluripotency assays currently used in the stem cell field to assess the pluripotency of your population? There are 2 ways to do that, teratoma assays and IBIS assays. Both the assays are not standardized, time-consuming, especially if you think at the teratoma assay, where you transplant your pluripotent stem cell population in a nude mouse and you wait for 30 days before to analyze the teratoma. For this reason, both the assays are not ideal to monitor routinely your iPSCs population. To monitor our pluripotent stem cell, we use the pluripotent functional identification assay. It is a standardized in vitro process to differentiate your iPS cells population in the 3 germ layers. It is faster to perform compared to the teratoma and the B assays and can be performed in a 24 and 96 well plates. The functional identification kit allows the differentiation of your iPSCs in the 3 germ layers in 3 days, and it provides antibodies lineage specific to quantify the differentiation of your cell. It provides OTX2 for ectoderm, SOC17 for endoderm, and brachyury for the mesoderm lineage. I would like now to give you an overview on how we use the pluripotent functional identification kit here at Bio-Techne. For example, we use it to monitor intercell line variability. In this first case, we have 2 iPS lines, iPSC3 and O29, and we differentiate them in the 3 germ layers using our functional kit. In this experiment, we use the Simple Western. After 3 days, we check for, in this case, the endoderm marker SOC17. And as you can see from the slide, both the cell lines are able to differentiate robustly in endoderm. In this other example, we used again the Simple Western technique. This time, we are looking for mesodermal markers nail. Both the cell lines, again, iPSC3 and O29, can differentiate in mesoderm very robustly, and as shown by the picture, they are able to express the markers NEO. Another way to monitor intercell line variability is using immunohistochemistry. Here at Bio-Techne we have an high-content instrument that allows us to quantify the antibody signal. In this slide, I show you the results about two other iPS cell lines in-house generated, IDLF1 and Fab2. The two cell lines can very nicely differentiate in endoderm and ectoderm. As you can see, more than 80% of the nuclei express SOC17 and OTX2. But when I differentiate the two cell lines in mesoderm lineage, there is an intercell line variability with the Fab2 cell line that express more brachyury than ADLF1. This table that I created using our in-house generated iPSCs cell line gives you an overview about the potential of the different cell lines to differentiate in the three germ layers. More plus sign you see, better is the differentiation obtained. I use the Pluripotent Functional Identification Kit to monitor intracell line variability as well. In few words, is the ability of different clones picked from the same cell line to differentiate in the three germ layers. In this particular experiment that I'm showing you, I picked three clones, 4, 1, and 2, derived from an in-house generated iPS cell line, KF. The three clones can very nicely differentiate in ectoderm, more than 70% of the nuclei are positive for OTX2, and in endoderm, more than 80% of the nuclei are positive for SOC17, as you can see from the diagram in this slide. In contrast, when I differentiated the three clones into the mesoderm lineage, I have observed very few nuclei positive for brachyury. Only in the KF1 clone, I have around 10% of the nuclei positive at brachyury. To test if the Wnt pathway is involved in the mesoderm formation, as is reported by many papers in literature, I try to activate the Wnt pathway using a small molecule to inhibit the GSK3 beta. As you can see, my data confirm the literature with a very nice increasing of brachyury expression when you add to the media the GSK3 inhibitor. In literature is reported that not only the mesoderm formation is affected by the Wnt pathway. In this slide, I want just to show you that without the GSK3 inhibitor, the level of SOC17 are very low during our mesoderm differentiation. So, using our kit, the SOC17 level are very low. But as soon as I add to the media that I use for the mesoderm differentiation the GSK3 inhibitor, I observe an increase of the SOC17 signal. So I confirm the Wnt pathway is not important only in mesoderm formation, but is involved in other like differentiation process as well. The Pluripotent Functional Identification Kit could be used to monitor variability in different cell culturing conditions. What do I mean about different cell culturing conditions? We know that the market of the media to maintain the cells in the pluripotent stage is crowded, and a lot of labs make their own media to grow and maintain their pluripotent cells. The Pluripotent Functional Identification Kit could be used as perfect tool to test if the media that you use to maintain your iPSCs let your cells, when it's removed, to differentiate in the three germ layers. I show you now some examples. I tested three different media for iPSCs maintenance on one cell line. In this first experiment, I performed an endoderm differentiation, and the cells grew in 3 different media, can differentiate in endoderm with more than 80% of nuclei positive for SOC17, as you can see from the images as from the graph. Unfortunately, I did not notice the same situation when I differentiate the cells in ectoderm. The cells maintained in media 1 can differentiate in endoderm better than the cells maintained in the media 2 and media 3. I observed the same situation with mesoderm differentiation, where the cells maintained in media 1 differentiate more robustly in mesoderm if compared with the other 2 media, as you can see from the Brachyury expression. To summarize this last part of the talk, I can say that the pluripotent functional identification kit could be a very good tool to use if you want to interrogate the pluripotency potential of your cells. As you can see from the cartoon, it's a standardized process and is fast to perform. In 3 days, you can get your results. We use standardized kit to differentiate the pluripotent stem cells in cardiomyocyte-like cells, hepatocytes-like cells, and neural progenitors-like cells. All the standardized kits have an antibody to confirm the differentiation of your cells. The STEM ex vivo cardiomyocyte differentiation kit, it is a standardized protocol to get beating cardiomyocyte in 8 to 12 days. You can find very nice beating videos on our internet site, but unfortunately on this platform the videos they don't work. Using the STEM ex vivo cardiomyocyte differentiation kit, you can get iPS-derived cardiomyocyte that express cell-specific markers as the cardiac troponin T in the picture on the left, and are responsive to the rhythmic calcium flux in the video on the right that doesn't work on this platform, but you can check it out on our internet site. Using the STEM ex vivo hepatocytes differentiation kit, you can get very nicely hepatocytes-like cells from iPSCs in 21 days. This protocol is a little bit longer compared to the cardiac one. At the end of the protocol, you can get very nicely differentiated cells that express hepatocyte-specific marker. As you can see from this picture, the cells express, for example, albumin, cytokeratin 18, HNF4-alpha, and AFP. Coming soon is another standardized differentiation protocol called the STEM ex vivo neural progenitor differentiation that allow you to differentiate the iPSCs in neural progenitor cells. In 7 days, you can get neural progenitor cells that express the marker PAC6 and SOX1, and they downregulate the pluripotency marker OCT4. Now we are working on downstream differentiation as well to get like a neural subtypes. In this case, from the picture, you can see motoneurons. I tested all these standardized differentiation protocol on all the cell lines that we are generated in-house here at the Bio-Techne. On this table, at the end of my presentation, I reported all the cell lines, again, that we have here in-house at Bio-Techne, and the differentiation capacity of each cell line in the 3 germ layers, and then in cardiac-like cells, hepatocyte-like cells, and neural-like cells. Again, more plus signs, better is the differentiation that I got. In summary, what I show you today is that colony selection is an important step to reduce variability, and variability between inter and intracell line exists. Another important aspect is that media that you use to culture your iPS cell line can affect the differentiation of your iPSCs. I show you some tool that we use here at Bio-Techne to reduce the variability during the colony selection as the live cell antibody staining and pluripotency marker expression, and to minimize variability during the differentiation as the functional identification kit or standardized protocol to differentiate your cells in cardiac, hepatocytes, and neural progenitors. I want to say thank you for listening, and now we will switch gear, and Tim will talk about the Gmp proteins.
Thanks very much, Fabrizio, and I appreciate the opportunity to talk to you today about Gmp proteins and their roles in cell therapy and regenerative medicine. Today I'd like to talk about growth factors, and we all know that they're an integral part of defined stem cell medias, and as a recombinant growth factor supplier, we realize that care has to be taken to manufacture proteins consistently. It's incredibly important to choose a supplier that minimizes risk and helps contain costs, and I'd like to talk to you a little bit about that today. So stem cell therapies and regenerative medicine therapies show tremendous promise. You can see there's news in the mainstream media on a regular basis, but we've all been waiting a long time for these things to make their way into the clinic. And it's basically, they're much different than traditional therapies, much different than, for instance, small molecules, much different than even biotherapeutics, and they're hard. This is an example of one of the more difficult forms of cell therapy, autologous cell therapy, where cells are removed from the patient. They then undergo some type of cell isolation or genetic engineering step. They undergo differentiation and expansion, and this is where Gmp proteins come into play. And then after that, then the cells themselves are reintroduced as the therapeutic. What makes this hard is there is inherent variability in biological systems. For instance, there's variability in cell populations from patient to patient that can make a particular therapy work different from patient to patient. There are associated costs in cell therapy and regenerative medicine that aren't seen in more traditional types of therapy, including extra training that's required and the costs of maintaining clean room facilities. There are still many manual processes that are still used too. For instance, you receive your cytokine in a vial in the mail, you dilute it, get it to the concentration that you're going to use, and then you use it. And these open manual processes create situations that can introduce risk. There are still questions about what these types of therapies are ultimately going to look like at scale. And because they're so new, the regulatory guidelines continue to be developed. And lastly, my focus today is the dependence on raw materials like growth factors that are themselves made in biological systems. And because of that, they also have an inherent variability. And they're also expensive. So I'm going to talk to you about some of the things that will help you choose the correct supplier and how that will actually go about minimizing the risk to your experiments and minimizing the risk to patients down the line as you move to marketing an actual therapeutic. And ultimately, minimizing the risks can actually help you manage costs in the long run. One thing that hasn't been clear is what the definition of Gmp is in this context, and so I'll focus a little bit on that and where the guidance for Gmp proteins comes from. And in my opinion, a high-level supplier should actually meet these guidelines and actually provide you more than the guidelines might suggest. First of all, the definition of Gmp ancillary or raw materials, as they're called in some places. Ancillary materials are materials that come into contact with the cell or tissue product during manufacturing but are not intended to be part of the final product formulation. So this means that they are used during the manufacturing process, used during cell culture, but ultimately they need to be washed away before the final therapeutic before the cells are used for the final therapeutic. And where does the guidance come from? One of the main places that the guidance comes from is from the U.S. Pharmacopoeia Convention, or the USP, and specifically Chapter 1043 on ancillary materials for cell and tissue-based products. So what Chapter 1043 does is it categorizes GMP materials based on their risk category. You can see at the top there is Tier 1, the low-risk, highly qualified materials. These are the therapeutic drugs or injectable materials that have the lowest risk. Tier 2 are low-risk, well-characterized materials produced in compliance with GMPs, and they're intended to be used as ancillary materials. This is where Gmp proteins fall. One thing I'd like to note is that suppliers of GMP materials aren't necessarily audited by the FDA, so the responsibility for ancillary material qualification resides with the developer or manufacturer of the cell or tissue engineered product. So this is a page that's taken from Chapter 1043. And it indicates some of the ancillary materials that fall under this Tier 2 risk category. This includes recombinant cytokines and growth factors, magnetic beads, monoclonal antibodies, stem cell media, things like that. In the far right-hand column is some indication of the qualification or risk reduction activities that should take place. Some examples, drug master files for cross-referencing, certificate of analysis, assessing lot-to-lot process performance, assessing removal from the final product because it's not therapeutic. Stability assessment as an ancillary material stored for use in manufacturing. And when relevant, confirmed certificate of analysis actually matches the specifications that you see when you do the experiments in your lab. And finally, you should always visit the vendor and see their facilities for yourself. But as I said earlier, this is probably just the minimum that you should expect from a supplier of GMP reagents. And there are a number of questions that you can ask, and choosing the right partners early is crucial and can significantly reduce costs that might include revalidation of your processes. So some obvious things that include qualifying vendors, include review of the literature and data to ensure the supplier has an established record in the industry, talking to your peers, making sure that asking if they've used the material and would they recommend it, and how is their relationship with that particular supplier. And then there are a couple of other questions that you might have that I think maybe I should spend a little bit more time on, and this includes what kind of quality system does that vendor have in place, and how do they control the quality of their raw materials? What kind of experience does the supplier have in the cell therapy space? And can the supplier scale when it comes time? So using R&D systems as an example, we have a very mature quality system. We're certified ISO 9001 and ISO 13485 for medical devices. And what this means is just that every process in the manufacturing of our GMP proteins is documented and traceable in order to make the same protein over and over again. This might include batch-to-batch consistency records, qualified vendors and material traceability. This could be incredibly important if you're interested in obtaining a cytokine or growth factor that is animal-free. We have to make sure that all of our materials that we use to manufacture that growth factor are animal-free as well. There is extensive employee training and documentation, equipment and maintenance and monitoring records. Stability assessments, for instance, we have controlled stability chambers where we look at the activity of our cytokines over time, and we have total confidence in the activity of our cytokines that's given on the C of A's that you receive. And finally, our quality assurance department signs off on all materials that get shipped from R&D systems. This all boils down to consistency, to making the same protein from the very first time you make it to potentially down the line when you are trying to market a therapeutic. And I'd like to give a shout out to our R&D systems quality team. There are 91 employees on our quality team out of about 730 that are at our Minneapolis facility, and this is actually a very high number when you consider the fact that R&D systems not only makes GMP level materials, but we have a large selection of research use only materials like antibodies, ELISAs, many different kinds of proteins as well. So experience, I think, is very important, and an experienced manufacturer can add value to the relationship. So some perfectly legitimate questions to ask are, how many clients do you have? How long have you been making a particular protein? For instance, R&D Systems has been making GMP proteins for 5 or 6 years, specifically TGF-beta as a research use only product for more than 30, and we have development manufacturing scientists here that know a lot about TGF-beta biochemistry and could talk to you in depth about that protein or any protein problem that you have interest in. Some other questions that you might ask: how often do you get audits? Do you have experience with clinical trials? And are drug master files available? And all of these will help you navigate the regulatory landscape. R&D Systems has many years of experience dealing with all of these and can help you work with the FDA and answer questions to move your therapies forward. And one thing that I would suggest that you do is start early with written inquiries. You can decide what the specifications that are most important to you for any given protein. Write those questions out and submit them to the quality department at a supplier, and they should be able to turn around the answers to those questions rapidly and give you an idea of whether there's even a starting point to move forward with that particular manufacturer. And what about the ability to scale? This is something that can be difficult with proteins. Yields can be very different among proteins, and so there are some questions that you should ask about capacity. You should ask questions about whether yields will support a successful therapeutic. Can you still meet specifications at a scale that would be required? These are incredibly important questions to ask, especially as you move a therapeutic through clinical trials and ultimately an accepted therapy. You might have to scale way up, and you might have a requirement for large amounts of growth factors, and an experienced company will be able to tell you what their ability is to scale up a particular growth factor. Some growth factors you can make in large amounts in batches, others are small amounts, and it's very important to know early on whether a particular supplier is going to meet your demands down the line. And how does scale affect cost? That's obviously an important thing to think about, and you should probably discuss those things earlier rather than later. Supply agreements and quality agreements. Talk about money early and find out what it's going to cost as you decide to scale up. And quality agreements, make sure that a supplier is going to be able to meet your specifications and your needs as they try to scale up a particular protein. One other thing I would suggest is starting with GMP materials as early as possible. You might find that costs are lower than you think, and it also reduces costs that might be associated with redevelopment when changing to a different protein or maybe even a different supplier. As I said earlier, proteins can be very different from supplier to supplier, and so using the same protein very early on and the same thing through clinical trials and ultimately, hopefully, to a therapeutic can reduce risk and headaches that can occur when trying to change your processes. Also, strongly think about having secondary suppliers. Again, proteins don't behave the same way sometimes from company to company, and if disaster happens and you need to move to a different company, you have a backup that you're confident in. And I'd like to just start to wrap it up here and say that Bio-Techne really has a focus on developing more than just vendor-customer relationships. We love to have people come and visit and sit down with our development scientists, our manufacturing scientists, our quality departments, and really learn a lot about what we have to offer. And you will find that they'll be able to answer even the toughest questions about your protein of interest. And so I strongly suggest that you come and visit. And so just in conclusion, hopefully I've told you a little bit about how choosing the right supplier can minimize risk and control long-term costs. Ask the right questions early and often. Experience counts. Choose a supplier that can add value to the relationship. And move to GMP as early as you possibly can in order to avoid changing materials in midstream. And thank you very much.
Thank you for that very insightful presentation. I hope our audience found it of great interest and value. If you do have any questions for Fabrizio or Tim, then please continue to submit them using the Q&A tool in the right-hand side of your screen. We've received a number of questions so far, so we'll begin to address these now live on air. The first question we have is for Fabrizio. Do you see differences in pluripotency based on the adult cell type that's reprogrammed, for example, fibroblast versus PBMC?
Yeah. No, if I have to be honest, I don't know. I do not observe differences in marker expression. As soon as if you get like nice colonies, I tried like different ways to interrogate the cells too. I tried like immunohistochemistry, flow cytometry, simple Western, and seems like that the pluripotency marker are well expressed in iPSCs derived from PBMCs and from fibroblasts.
Fantastic, thank you. The next question is for Tim. What if I'm working on cell A therapy project and GMP isn't available? What are my options?
Well, there is no regulation that says that you have to use GMP for cell therapies. So if you have a really good idea of what your specifications are, you can sit down with a particular vendor and go over their research use materials and see if they might be satisfactory for you. And it's good to get the regulatory body like the FDA involved early and make sure that they're on board with a particular raw material if it's not GMP. And lastly, another option is to talk to a company that can turn one of their research use only proteins into a GMP protein, and we actually have these types of custom agreements happen fairly regularly here at R&D Systems, so that's an option too.
Lovely. Again for Fabrizio, after the live stain for the iPSC picking, do you use a cutter to pick out the colony, or do you use a cutter or a scalpel? In your experience, which method picks up a clean colony best?
So in my experience, it's about how clean is the colony that I'm going to pick. So sometimes you can have like colonies that are very clear, that express beautifully the pluripotency marker. So in that case, I just pick them using a pipette, a 200 microliter pipette with the tip. In some other cases, you can get like colonies surrounded from like differentiated cells. So in that case, I tried with a cutter to remove all the differentiated cells and then pick just the undifferentiated cells. Sometimes it's like I have some differentiated cells during the passaging, but I lose them passaging the iPSCs. The best thing for me is like you have to test your iPSCs for pluripotency marker every like 5-10 passages, and be sure that like you have a very nice, good expression of pluripotency marker as OCT4 more than like 95%, SOX2, NANOG, and these kind of markers.
Great. The next question is also for you, Fabrizio. Do you have anything for mesenchymal stem cells?
Yeah, we have something for mesenchymal stem cells. For sure, we have like antibodies, we have like growth factors, small molecules, we have like a kit to differentiate mesenchymal stem cell in the lineages, like I think chondrocyte, osteoblast, and I'm not sure if we have other kits, but it's like if you go on the internet site, on the Bio-Techne, and you type in like mesenchymal stem cells, you can find all the products that we have for that cells. My field is like more inducible pluripotent stem cells, but we have tools, kits, standardized kits for differentiation and antibodies for mesenchymal stem cells as well. Hello?
Lovely, thanks, Fabrizio.
Sorry.
Next question is for both of you, or either of you. Do you sell iPSC lines introduced during your presentation?
Do I sell iPSC lines?
Yeah, do you sell the iPS cell lines?
No, we don't sell iPSC lines. We just generate in-house iPSC lines using Sendai virus, and we just use in-house to test our products and to like develop it to differentiate them in the different like lineages.
Great. Tim, how much detail can you share about your manufacturing protocols to make sure they meet the needs of our application?
Well, it might be variable from protein to protein, but this is where the audits come into play, and you can sit down with us and we'll tell you as much as we possibly can about you know, a given process. But this is also where the drug master files will come into play, and any company should be able to submit drug master files to, for instance, the FDA, and the FDA would have access to any types of confidential information to ensure that they're producing a raw material that doesn't raise any red flags.
Great, thank you. Fabrizio, you mentioned about neurodifferentiation kits for iPSCs in your presentation. We have someone asking for more product information, so I guess we'll address that offline.
Yeah, that kit is coming soon, so it's not released yet. I can say that is a kit to differentiate the human inducible pluripotent stem cell in neural progenitor cells. And you can get very nicely differentiated cells between 7 and 10 days, but we didn't release yet the kit. So as soon as we be in the market, we will like give more information about that.
Great, we'll look forward.
And I will send more information as well. Yeah.
Yeah. Again, for you, Fabrizio, what equipment do you need to start a laboratory unit for iPSCs?
What equipment? You need a lot of things. You need for sure hoods. You need like incubators, centrifuge. You need like a clean space, very clean. How can I say? You need a lot of money. You need a lot of things. It's not that easy like a question. I can talk about this for like 1 hour if I have to be honest. But summarize. You need experience. You need like good people that are like hard worker, because with the iPSCs you have to spend a lot of time with them. You have to change media almost every day. You have to work during the weekends. You need like cell culture hoods. You need incubators. You need like all the equipment for a cell culture lab, and a lot of passion. Yeah.
Lovely, thank you. Fabrizio, again, you spoke about immunohistochemistry and Western blot for iPS cell validation. Is flow cytometry a viable option as well?
Oh, flow cytometry is a very good and is a viable option as well. Yeah, a lot of people, they use flow cytometry in the field, and I think it's one of the best options to regularly monitor your iPSCs. So yeah, definitely yes, it's a good option.
Great. What is the clear difference between products from DNA cell lines and iPSCs?
Oh, can you repeat the question? Sorry, what?
Sorry. What is the clear difference between products from DNA cell lines and iPSCs?
Products from DNA cell lines. I
Yeah.
don't understand the question.
No worries, we'll move on. Might you have differentiation medium for retinal pigment epithelial cells as well? It's a derivative of neuroprogenitor cells for our eyes.
I think it's like I don't know, I have to check if we have differentiation media for retinal pigment epithelia. I don't think so, but I'm not sure. I should check on our internet site. It's like the neural differentiation is a new project. The neural differentiation, like progenitor cells from iPSCs, is a new project. So it's like we are developing stuff right now. We are developing media. We are developing kit to differentiate the iPSCs in this kind of lineage. But as I said, is a new things. So I think we don't have right now a media to differentiate the retinal pigment epithelia. But you can check on our internet site under the stem cell and see if there are some tools that you can use in your research.
Great. Question for both of you. What method or methods does Bio-Techne use for cell reprogramming?
So we use, as I said before, Sendai virus. So we have Sendai virus for cell reprogramming, and it works very, very nicely. So we are stuck with that. But as you know, you have like a lot of different techniques that you can use to reprogram the somatic cells.
Great, thank you.
No problem.
Another question. Do you have weekend-free culture medium for iPSC expansion?
Sorry, can you repeat the question?
Weekend-free culture medium.
Weekend free?
Yes.
The weekend free, sorry.
Yeah, sorry.
Sorry, I didn't check. Right now, we don't have a culture media for iPSCs that we sell. So I know that there are some medias that are weekend free that are out there. But here, like Bio-Techne, we don't have our stem cell media. So it's like I say no. But there are some media that can be like weekends ends off.
Great. We have a question for Tim now. Tim, are synthetic components of cell cultureware considered ancillary materials for cell products?
It's not listed in the 1043 chapter, but if there are certain specifications that you have regarding cell cultureware, you should make sure that a company can answer any of those questions for you. As I mentioned earlier, there are no strict guidelines for these, and a lot of the responsibility lies on the manufacturer of the cell therapy.
Great, thank you. Thanks very much, Tim and Fabrizio, for answering those questions for us today. We have one final question. A non-xeno-free media Gmp?
They can be. So, as I said, what we have for Gmp ancillary materials are regulatory guidelines. You can make a Gmp material that is not, for instance, animal-free. It'll have a lot of documentation, a lot of traceability associated with it, but you'll have to decide whether having an animal-free protein is critical for your application or not. But yes, you can have a GMP material that's not xeno-free.
Thanks. Again, thank you for answering all those questions. Before we wrap up, do you have any last comments you'd like to add?
I don't. I'm satisfied.
Great.
I'd just like to say that I really appreciate the opportunity to talk to everybody today, and look forward to some of the feedback and answering some of the questions that we actually didn't get to today.
Well, we really appreciate you taking the time to talk to us. I'd like to once again thank our wonderful presenters, Fabrizio and Tim, as well as our listeners for your time and for your questions. If you have a minute, we'd be really grateful if you'd complete our feedback survey, which you can find in the resource section of your screen. Remember, you'll find a recording of this webinar available to view on demand on RegMedNet at www.regmednet.com/channels/196-webinars. It will be available shortly, and you can also find details of our upcoming webinars. Thank you again for attending, and goodbye.
Thank you.